Hello everyone, my name is Thomas Cowgill. I am a master's student at the University of Edinburgh, and today I'm going to be talking about my work on metriorhynchoid crocodile. Now, metriorhynchoids, these are a group of marine crocodylomorphs spanning from the early Jurassic to the early Cretaceous, and these include the only crocodiles, and indeed the only archosaurs, fully specialized for pelagic marine life, the metriorhynchids. Now, these crocodiles have several adaptations for marine life, including a hypercircled tail fin, paddle-like limbs, loss of osteoderms, making them more streamlined, orbits on the side of their skulls, and there's even evidence that they had salt glands. Now, unfortunately, we don't know very much about these crocodiles, but only recently we're starting to understand their paleobiology, including their endocranial anatomy. Natural endocasts have shown that preserved evidence of salt glands, and advances in CT scanning have allowed us to take a closer look at their brains and inner ear. However, one aspect of endocranial anatomy that has often been neglected is the paranasal sinus system. Now, paranasal sinuses are a network of air-filled diverticula in the facial skeleton, in the snout, connected to the nasal passage. These are present in both mammals and archosaurs, including crocodiles. And living crocodiles, like this Cuban crocodile you can see here, exhibit a complex array of paranasal sinuses. Now, taking a closer look at this crocodile, you can see several different sinuses connected to either the nasal cavity or the nasopharyngeal duct, the tube connecting the nasal cavity throat. Now, and while modern crocodiles have several different sinuses, only one is shared between them, and indeed they share with all other archosaurs, the antorbital sinus, here in green. Now, what's interesting about this sinus in modern crocodiles is, unlike other archosaurs, like this Tyrannosaurus here, the antorbital sinus is bordered by a large opening in the skull called the antorbital fenestra. Modern crocodiles don't have this fenestra, so their sinus system is completely internalized. Previous work on metriorhynchoids has shown some of them have reduced paranasal sinuses, and this makes sense. These are marine animals, and having a large pocket of air in your skull is going to affect your buoyancy. However, the system has only been studied in very few metriorhynchoids. I think there's only been 2 species where it's been studied. And this limited data means we have no understanding of the system variation in metriorhynchoids, what function, if any, it performed, and how the system evolved. Our work begins to address this problem by studying the paranasal sinus system and associated pneumatic structures, i.e. the nasal cavity and nasopharyngeal ducts, in multiple metriorhynchoid species. First, we established a system in CT scans of extant longirostrine crocodiles, so crocodiles with a long tubular snout. We use these as a comparison because they have the most similar skull morphology to metriorhynchoids. Then we look at CT scans of well-preserved metriorhynchoid skulls to identify evolutionary trends. Here is our sample. We have 10 specimens across 8 species. 2 species of modern longirostrine crocodile, the gharial and false gharial, and we used both adult and juvenile specimens, 2 basal metriorhynchoids, Pelagosaurus and Eoneustes, and 4 derived pelagic metriorhynchids, Metriorhynchus, Pricosaurus orcanensis, Pricosaurus schroederi, and Torvaneustes. So, taking a look at our modern crocodiles, they have surprisingly extensive sinuses, more extensive than we thought they would be, with both adult specimens showing multiple pairs of well-developed sinuses. In the juvenile and subadult specimens, however, these sinuses are much more reduced. You can hardly see them in these images here. So, taking a closer look at the juvenile and subadult, you can see we could only identify the antorbital sinus. And this sinus opens posteriorly into the nasal cavity. It is always ventral to the dorsal alveolar canal, which you can see in red. And in the false gharial, you can see in the bottom right, it actually shares an opening into the nasal cavity with the dorsal alveolar canal. They share the same cavity. Looking at our metriorhynchoids, our most basal species, Pelagosaurus typus, we could only find the antorbital sinus on the left side. You can see in this cross-section in the bottom left, there's a small dividing wall within this cavity, and both the antorbital sinus and dorsal alveolar canal share its cavity. Now, compared to adult longirostrine crocodiles, this system is much more reduced. However, compared to our juvenile longirostrine crocodiles, the systems are actually very similar. You can see they all have reduced antorbital sinuses. The sinus always opens posteriorly into the nasal cavity. It is always ventral to the dorsal alveolar canal, and in both Pelagosaurus and the false gharial, the antorbital sinus and dorsal alveolar canal share the same cavity. Now, there are two ways of looking at this. Either this is evidence of pedimorphosis in metriorhynchoids, or this could represent a basal condition in all crocodylomorphs. In our other basal metriorhynchoid, Eonoustes, we couldn't find an antorbital cavity, so we haven't segmented an antorbital sinus. However, because the antorbital sinus is present in both the more basal Palagosaurus and in the more derived metriorhynchids, which we'll see later, we can be confidently infer that the system is present in Eonoustes. And if it is, as you can see in this cross-section, it's most likely sharing the same cavity as the dorsal alveolar canal. The derived metriorhynchids have a very different sinus system, and it's best observed in our two Cricosaurus species. Here, the antorbital sinus has a conical morphology, and most interestingly, the antorbital cavity actually opens into the orbit through a large postnasal fenestra, which you can see in the bottom right here. Now, we know that in living birds, the antorbital sinus also extends into the orbit, creating an accessory suborbital sinus. And we believe the same thing is happening here in metriorhynchids. And if so, this would be unique. No other crocodile group has an accessory suborbital sinus. You can better see this in Cricosaurus schroederi. Here you can see the suborbital sinus in purple extending through the postnasal fenestra into the orbit. In our other metriorhynchid species, the sinus system is less well preserved. In Metriorhynchus, we also have evidence for a suborbital sinus, but it's not as well preserved. And in Torvanustes, we can only segment part of the antorbital sinus, though also showing a conical morphology. So we see that the suborbital sinus was not only present, but widespread among Metriorhynchids. However, we don't know how extensive this sinus was. The suborbital sinus is not like other paranasal sinuses. It doesn't invade the skull bone, so it doesn't have a cavity where we can measure its volume. However, we also don't know how extensive this sinus was through the postnasal fenestra it's traveling through, because other systems go through this fenestra as well, like the jaw muscle. The dorsal pterygoides muscle is also known to extend through the postnasal fenestra in archosaurs, like in this Palagosaurus muscle reconstruction you can see here. So we have two hypotheses for what's going on through this fenestra. Either the suborbital sinus is dominant and the pterygoides muscle is reduced compared to other crocodylomorphs, or the pterygoides muscle is dominant and the suborbital sinus only occupies a small area. We also don't know what possible function this suborbital sinus performed. However, looking at modern birds and other marine animals, we've come up with two hypotheses for the most likely possible function. In birds, because the suborbital sinus is located right next to the pterygoides muscle, when this muscle contracts, it compresses the suborbital sinus, which actively ventilates the sinus system. And we believe the same thing was likely to have happened in metriorhynchids, so this could have performed a respiratory function. The alternative is this performed a diving function. In modern whales, like this dolphin, they have lost their paranasal sinuses. However, instead, they have a system of paranasal air sacs outside the skull, and these expand and contract during changing water pressures when diving. And the suborbital sinus in metriorhynchids could have done the same thing. So when the metriorhynchid dived, the suborbital sinus would retract through the postnasal fenestra. When it returned to the surface, it would expand through again. However, at this stage, we don't exactly know what's going on here. We also looked at the nasopharyngeal ducts, and in modern crocodiles and palagosaurs, they both have very similar morphology, roughly the same cross-sectional area along their length. But in the derived metriorhynchids, these ducts are dorsoventrally expanded. You can see in these cross-sections here, they are much taller, and the palatin bones below these ducts are also much thicker. And we believe this will allow for stronger ventilation at the water surface, so they could take in bigger breaths before diving. However, this also assumes they had expanded lung capacity, which we don't know about at this time. Finally, we looked at the nasal cavity, and like with the nasopharyngeal ducts, in modern crocodiles and pelagosaurs, there's a very similar morphology. However, in Eonoustes and the metriorhynchoids at the posterior end of the snout, the nasal cavity is dorsolaterally expanded. And interestingly, this expansion only occurs in species where salt glands have been inferred. So this was likely an adaptation to house these enlarged salt glands. So our work has revealed a lot about the paranasal sinuses of metriorhynchoids. However, we still don't know the full story about their evolution, and there are several lines of evidence we still need to go down to understand the full picture. First is teleosauroids. These are another group of Jurassic marine crocodiles, the sister group of metriorhynchoids. An understanding of paranasal sinuses in these crocodiles will help us understand the basal condition of both groups. We can also look at even more basal crocodylomorphs, sphenopsuchians and protosuchians. And this will enable us to understand the basal condition across all crocodylomorphs. And finally, we can perform muscle reconstructions on metriorhynchoids, understanding how the dorsal pterygoides muscle extends through the postnasal fenestra. Understanding the full extent of this muscle will let us test for the presence and extent of a suborbital sinus. Thank you very much for your time. Any questions?